So here we're going to focus more on what exactly happens with the chromosomes during meiosis. A lot of it I mentioned before, but let's just go through it step by step because it is the most important part of understanding what happens with meiosis. So very much like with anything else, we're just going to start with the way a normal cell has a homologous pair. And in this one, I'm just creating something of an example. So let's say that this homologous pair, the purple one's from the mother and the blue one's from the father. And let's say amongst many other things, it codes for the eye color and it codes for whether someone is tall and short. You'll find that most genetic structures are actually more complicated than just this, but let's just assume it's like this. Okay, so as you can see on these structures, the gene for height and the gene for eye color are sort of right next to each other in terms of where they are mapped. They just code differently. So these are called alleles, these pairs of genes. And let's say the mother's allele has blue eyes and is tall, whereas the father's one is brown eyes and short. Okay. So during interphase, that's when the DNA replication occurs. So basically it just makes an exact copy of each chromosome. So you can see the maternal chromosome, it's still the same. Both of the maternal alleles are tall, and both of the maternal eye color alleles are blue, and both of the maternal height alleles are tall. And the same for the father, they're both still brown, they're both still short, because these are still the exact replicas that happen in interphase. Okay, so now we get to the important bit during prophase 1, where there's crossing over. So again, it's usually a lot more complicated. These things cross over and over, all over both of them, but we're just doing something to indicate exactly what sort of thing could happen. So let's say that our homologous pairs cross over. One arm crosses over at the area where the height is, and that place that's crossing over is called the chiasmata. So that's what happens in prophase 1, and where it crosses over, they actually swap the genetic material that's there. Okay. So during metaphase and anaphase 1, what happened is that the chromosomes are separated, but you can now see that they actually have different information. So this one is blue, tall, but now also short, whereas this one is brown, short, but now also tall. Oh my gosh, I wrote that wrong. This is supposed to be tall. Oh, let me find my actual pen. Where is this? Oh, here's one. Sorry about that. So, this here is supposed to say tall because it comes from the mother. Tall. Okay. All right. Anyway. Yeah, so basically you've now got your homologous chromosomes, but their copies are no longer genetically identical because they've switched information. Okay, and then at this point these two get pulled apart. So this chromosome goes to one side, this chromosome goes the other side, and they are each going to go into a separate cell. And remember, since they are no longer homologous pairs, they're simply inexact copies of each other. These are now haploid cells. And then thereafter, nothing much happens with the DNA during telophase 1 cytokinesis, and prophase 2, it's more or less the same as normal. Okay, then during metaphase and anaphase 2, at this point, the chromatids are now separated. So these copies are sort of separated from themselves. So here you've got a blue and tall copy going this way. Here you've got a brown and tall copy going this way. Here you've got a blue and short copy going this way. And here you've got a brown and short copy going this way. And each of those is going to go into its own haploid cell, which is a sex cell. Okay. So basically what you can see is that you've now got 4 different haploid cells, each coding for a different type of human. So this one is coding for a human who's tall and has blue eyes. This one is coding for someone who's short and has blue eyes. This one is coding for someone who's tall and has brown eyes. And this one's coding for someone who's short and has brown eyes. And so that's the most important thing to keep track of what's going on during meiotic division. Okay. And then finally, I just did a table here that we can talk through very briefly. But the idea is just to sort of compare the most important differences between mitosis, meiosis 1, and meiosis 2. There is another table that I've sent along with the practice questions, but it's a little different. It's got a lot of information. I'm just trying to hit the sort of the big points so that you can sort of follow. And for this, I think it's nice to put these pictures side by side so that you can kind of compare the two. Okay, so I'm not going to talk through each of them now. We can do that together, but what I'd recommend you do is that you put your papers next to each other and then just look at each one. So for example, what is each cell type starting as? So mitosis is starting with a single diploid cell. Meiosis 1 starts as a single diploid cell, but meiosis 2 starts with 2 haploid cells. Okay. And then what are the starting chromosomes? For mitosis 1, it's a homologous pair. For meiosis 1, it's a homologous pair. For meiosis 2, they are unpaired. And so on. And then you can just go over. I didn't fill in everything. For instance, there's nothing particularly noticeable that happens during mitosis or meiosis 2 during prophase. The only thing that you have to really remember is special is that during meiosis 1, there's the crossing over and then the independent assortment and everything. Yeah, so I think that can be quite handy as a just looking at the division in a slightly different way.